Good afternoon friends, myself Syed Mohammed Quatadah, Assistant Professor, Genetics and Plant Breeding, Faculty of Agricultural Sciences and Allied Industries, Rama University, Kanpur. Today I am going to discuss a very important topic, expression of gene in eukaryotes. Actually, as we know, the genes itself does not produce their characters, it always needs some factors to express, and these genes express their characters in the form of protein, in the form of hormones, in the form of enzymes. So, as you know, the DNA is present inside the nucleus of the cell, inside the, for example, this is the plant cell, okay, and this is the nucleus. The DNA is present inside the nucleus of the cell. Huh? These DNA are double stranded. These DNA are double stranded and they have the specific sequence of nitrogenous bases, a specific sequence of nitrogenous bases which are in a sequence, which are in the sequence, and this specific sequence of nitrogenous bases is called the gene, is called the gene. So, these expression of the gene completes in two steps. First one is the transcription, and second one is the translation. Actually, in transcription, DNA produces mRNA, and this process is called transcription, and from mRNA protein, enzyme, etc. are produced, and this process is called translation. This process is called translation. Means transcription and translation, these are the two methods through which DNA the having information encoded sent to the nucleus, and in the from the nucleus to the cytoplasm, and where this encoded information is being translated in the form of protein, and in this way the character expressed. So, this is the eukaryotic transcription overview, in which you may say the first step in the transcription in eukaryotes is the initiation. First one is the initiation, then elongation, and this one is the termination. These are the three basic steps in which the transcription process completes. Like initiation, through that initiation, through the process of initiation, those sequence of DNA which is being copied and mRNA should be formed. In this process, the sequence should be identified in the DNA, where the transcription process has to be start, and after that in that place a promoters recognize the initiation, transcription initiation positions, and this position is the Tata box which is present on the DNA. This is the Tata box, TA sequence. This is the called Tata box, TA sequence. This Tata box is the actually transcription initiation position in the eukaryotic DNA. In this place, a substrate molecule binds. These are the promoters. These are the promoters. Actually, promoter is that this is the factors which binds to this place which binds to village, and it helps in associating the transcriptional binding protein TBP, transcriptional binding proteins. Transcriptional binding proteins have that different, and in that RNA polymerase have the different factors which is called RNA polymerase RFP factors. These are the factors. When these factors along with the RNA polymerase enzymes binds to this initiation factor Tata box, then the initiation process starts, and due to this helicase activity of the polymerase second enzyme, these bonds break between these two double helical strand. One strand is used template enzyme, and the transcription of the mRNA starts in the 5' to 3' directions. After that, elongation process takes place, and lastly, when the information present on the DNA molecule is being copied in the form of mRNA, then the chain terminates, and this mRNA is transferred from the nucleus to the cytoplasm. This is the double stranded DNA molecule, 3', 5' and 5' to 3'. Actually, this is the place where Tata box is present, TA. This Tata box is the transcription initiation position in double stranded DNA molecule in case of the eukaryotic DNA. Then after that, after recognizing the promoters bonds, transcription binding proteins associated with these factors, and helps to bind the promoters and stabilize the TBP, transcription associated factors. After that, these are the different transcriptional factors. Transcription factors A stabilize the DNA to transcriptional binding proteins to promoters. Similarly, transcriptional factor second F, it helps in bind tightly to RNA second, transcription second B, and prevents binding of RNA polymerase second to non-specific DNA sequence. मतलब इसका काम क्या है? These are the factors. It prevents the transcriptional factor to bind to that place where transcriptions is not being started. And third one is the transcription factor second B. It recruits the RNA polymerase second transcriptional factors cocomplex, and this is the RNA polymerase second enzymes. This whole concept enzyme is called the holoenzymes. Okay? And this is the transcription factor second H. It have the unwinding and helicase activity, unwinds the DNA at promoter site, where the promoter site binded to this place. This is the Tata box, and promoters bind, and helicase activity means breaks the double stranded DNA molecules नाइट्रोजनस बेसिस को ब्रेक करने में ये मदद करता. फॉस्फेटेज RNA polymerase second activity, recruit the nucleotide excision repair proteins. So in this way, the whole holoenzymes means RNA polymerase enzymes, है ना, through with the RNA polymerase enzymes, promoters, transcriptional binding proteins, and their transcriptional factors, associated factors. These includes together and starts the chain initiation activity. And as you know, the double stranded DNA molecules DNA molecules have the nucleotide sequence adenine, guanine, cytosine, thymine, adenine, cytosine, guanine, adenine and thymine. But in case of RNA, in place of thymine, there is uracil. Similarly here uracil means these should be adenine uracil, guanine cytosine, adenine uracil, adenine. These are the sequence. So in this way the mRNA 5' to 3', mRNA always forms and copies this sequence as a template, mRNA. After maturation, this mRNA is transferred from the nucleus to the cytoplasm. But you know, this mRNA molecule is yet not ready for the translation process, because these RNA molecules have many uninterrupted sequence of the nucleotide which will not be transcribed or read. So after that mature mRNA is formed, mature mRNA. This process is called splicing. Of mRNA. Means this mRNA have the coding and non-coding sequence in the uninterrupted form. So to make the mature mRNA, the uninterrupted means non-coding sequence from this mRNA will be eliminated through the helicase activity. After that only the coding sequence of the RNA, which will convert into the protein through the process of translation, is formed, and this process is called the translation. So in this way, in the transcription process, first of all double stranded DNA molecules binds with the promoters, and promoters always binds to that place of the DNA molecules where TA sequence is present, and this TA sequence is being recognized with the transcriptional binding proteins. After that, these transcriptional binding proteins along with their cofactors like transcriptional factor second A, transcriptional factors F, transcriptional factors B, transcriptional factors H, transcriptional factors D. These are the different transcriptional factors, and each transcriptional cofactors have the different activities. These together bind along with the RNA polymerase enzymes and breaks the double stranded DNA molecules nitrogenous bases. After that, they use one of these strands as a template and copy the nucleotide sequences. But as we know, in the RNA in place of thymine, uracil is present. So the mRNA have the adenine, guanine, uracil in this way, means adenine, guanine, uracil nitrogenous bases sequences, and these sequence have the encoded information as the double stranded DNA molecules have. After that, this encoded copy of the message transfer from the nucleus to the cytoplasm of the cell, and in the cytoplasm of the cell, this mRNA molecule, for example, this is the mRNA molecule attached with the ribosomes, and you know each ribosomes have the two site, one is the P site and another one is the A site. P site means the peptidal sites, peptidal sites. The tRNA along with tRNA helps in transferring the nucleotide sequence, adenine, uracil, guanine, cytosine. These are the nucleotides. The tRNA transferred to the peptidal sites, and in this way these specific nucleotide sequence U, AUU, UUA, GUG, GGA, CCA, these are the sequence of nucleotides, and each triplet sequence of the nucleotides has the specific express the specific amino acids, and these peptidal sites adds this nitrogenous basis sequence from the P sites, and this is the assimilation site, acyl site, means convert this nucleotide sequence into the specific amino acids, and these amino acids polypeptide chain. This amino acids polypeptide chain is called the protein. So, in this way the entire process of transcription and translation completes in the eukaryotic organisms. Means, as we know that gene will not produce their character sequence. Gene always needs some factors, and the factors is being associated with the production of enzyme, proteins or hormones. In this way they express in the eukaryotic cells responsible for the expression of the characters. Thank you.